It's my great privilege to introduce our keynote speaker, Dr. Eve Marder. Dr. Marder is the Victor and Gwendolyn Bainfield Professor of Neuroscience at Brandeis University. Dr. Marder's research on small neural circuits found in crabs and lobsters has revolutionized our understanding of the fundamental nature of neuronal circuit operation, including how neuromodulators control behavioral outputs, and how stability of circuits is maintained over time. Dr. Marder is a member of the U.S. National Academy of Sciences and a fellow of the American Academy of Arts and Sciences. In 2016, Dr. Marder was recognized for her contributions to neuroscience with the Kavli Prize. And in 2023, she was awarded the National Medal of Science from President Biden, the highest honor for scientists in our country. Dr. Marder, to me, is a force of nature. As an early graduate student, her work on understanding how neuromodulators influence circuit output terrified me and also inspired me because it revealed an awe-inspiring complexity that I thought I could never understand. And many years later, Eve continues to inspire me, and her foray into physiological response to climate change initiated much of the work that drives my day-to-day life. So please join me in welcoming Dr. Marder.
I'd like to thank Angie and all the other sponsors for this meeting for the opportunity to speak. And I would like to say that preparing this talk was really hard, and you'll see why in a moment. It was really hard for the following reason. These are the wonderful people I've worked with, and I've put in green all the people who are my present and former trainees who've done work that's actually relevant to this meeting and to a lot of the issues we're going to be talking about. So I sat there trying to figure out which subset of all the miraculous work that they have done I could talk about, and I went into paralysis. So I really did. And my solution to the paralysis was to highlight everybody's names in green and to point out that this way of thinking has really percolated and sort of dominated a lot of the work going on in my lab for the last sort of 15 or so years, maybe a little bit longer. And therefore, I apologize to everyone whose data I'm not showing and hope that you'll forgive me and still be my friend. Okay, and they're all wonderful. So, this all started with our understanding of degeneracy. What we call degeneracy is that there are multiple mechanisms that can give rise to very similar behavior. And this can be seen in both experimental and computational work. And you'll see that our first understanding of this became a point, a jumping off point, that actually ended up in what we're doing today. And I started out with a very basic fundamental question, which is how different could different nervous systems be in the same species and still generate similar characteristic behavior? That is to say, how different can two crabs be in the organization of the nervous systems and still be successful crabs in the ocean? Or how different can your respiratory centers be from each other and still have you be breathing? And so that was the problem that started driving us, and it ended up with us worrying about what's going on with the lobsters and crabs in the North Atlantic Ocean, and you'll see how that came about. But it was a very simple path. And I bring this up because I really believe that curiosity-driven basic science leads all of us into new directions, and those are directions that may be far more profitable than ones where you set out to say, I'm going to do X, I'm going to solve cancer. You may end up solving cancer because you were chasing something else. So, I've worked my entire scientific career on the crustacean systematic gastric nervous system. I started out as a graduate student at Al Silverson's lab at UCSG, and we kept our animals down at Scripps, and we bought them from local fishermen who went out in their boats and brought back, at that time it was Panularis interruptus, And we kept them in running seawater down in scripts, and we'd go down, and I'd bring one back in a brown garbage bag, and that would be the experiment of the day. So I'm very partial, Penny-Lars interruptus. I did at one point try and keep working on them, but they had to be shipped in from California, and we had to go get them from the airport, and they came in boxes, and the guys in the air freight were terrified of them, so we stopped. It was very. But the stomachogastric nervous system was a very important nervous system in those early years because it's one of the premier examples of a nervous system which could generate fictive motor patterns when the nervous system was isolated from the animal that resembled very closely the motor patterns generated in the animal. So the animal that most of the data I'm going to show you today is Cancer borealis. Cancer borealis, the way we study them, they're about so big, and they're 5 to 7 years old, and they've spent their entire life in the North Atlantic. And this is really important because in the North Atlantic, they've dealt with all the vicissitudes of the weather and the changing tides, etc. Now, what I used to do, my wonderful lab now does, we start with a crab, and the stomach is up there. And I don't know if I have a pointer. Is there a pointer? No, there's no pointer. So I'm just going to gesticulate as if you can read my mind. Okay? That you can all read my mind, right? That yellow circle is where the stomach is, underneath the heart carapace. And to some extent, it's been possible to get recordings in vivo in spite of the heart carapace. And Alan Wolfenstein, who's here, is an expert at getting brilliant recordings in vivo. But in general, what we do is we take the stomach out, and that's a picture of a lobster stomach. I'm sorry, but you never know what happens when people start to make slides from images in the web. Anyway, the stomach has 4 major regions. There's the esophagus, food enters there, and then there's an area called the cardiac sac where it's stored, and then there's gastric mill region, which has the muscles involved moving a bunch of teeth inside the stomach, and the pylorus is at the back end of the stomach, which is a filtering device. So there are actually 4 rhythms that are generated by this nervous system, but we're going to only be talking about 2 of them, the gastric rhythm and the pyloric rhythm. Now, here's the stomach with the nervous system attached, and what the brilliant people, thank you. What the brilliant people in my lab do is they sit under a microscope and they dissect all these nerves off the surface of the stomach, and they lay them out in a Silgard-lined petri dish, usually about so big, at which point it can be superfused with physiological saline. You can change the composition of the saline. The stomatogastric ganglion is desheathed, usually, and can be used for intracellular recordings, and extracellular recordings can be placed on the motor nerves. So we can simultaneously, and I laugh when I talk to vertebrate people who say, oh yeah, we can record from 3 neurons simultaneously, and I laugh because a good STG physiology can actually record from all 30 neurons or all 26 neurons in the stomatogastric nervous system simultaneously using a mixture of intracellular and extracellular recordings. Now, here are some recordings of Sonal Kedja, who's here, made of the major components of the pyloric system. Here's an LP neuron, a PY neuron, and a PD neuron. And those are intracellular recordings from the cell bodies. And they are identified according to action potentials in the motor nerves. And those are then identified in principle because of which muscle they target to. So these neurons are absolutely unambiguously identifiable in terms of their targets. And therefore, this becomes very important because you don't have to be hostage to looking at activity patterns or patterns of synaptic inputs to really identify the cells. Okay, this is a triphasic motor pattern, LP, PY, PD. LP and PY innervate constrictor muscles for the pylorus, and the PD innervates the dilator muscle. So the rhythm is constrictor 1, constrictor 2, dilator, constrictor 1, constrictor 2, dilator. Now, the important thing for a lot of what we do is that this rhythm persists continuously in the animal, and much like your respiratory rhythm, which also continues throughout your lifetime in the animal, that property of the rhythm has made it very, very useful as a monitor for what's happening. This is what used to be called a wiring diagram or a connectivity diagram. Today it's called a connectome, and I always laugh because, and I think you guys will all understand, we had this connectivity diagram or connectome in 1980, and that only meant possible that it was the beginning of a lot of very interesting questions about how it works. And so I just often say to people, the connectivity diagram or the connectome is the beginning, not the end of understanding how it works, assuming that everybody here wants to understand how it works. I never understood what the it was, but that's okay. Okay, so that's the fast pyloric rhythm. Notice this is 1 second, and the canonical period of the pyloric rhythm is about 1 hertz. However, these recordings, and we seem to always be missing a time base, and it doesn't appear. It's been missing for 25 years, and it just does not appear. I don't know why it doesn't fix itself, but it doesn't. So these recordings, I believe, I think they were from Jim Wyman many years ago. But you can see this is an intracellular recording from a neuron called LG. That's the lateral gastric. You'll hear about this neuron later. And this is a snare and a delusion. Notice that the LG depolarizes, fires a long burst of action potentials, hyperpolarizes. During the time that it's off, it's getting rhythmic inputs in time with the pyloric rhythm. Now, the gastric rhythm can be seen as alternations from the LG and then either the DG or the GM neurons, which are shown on the extracellular recordings up there on the DGN. At the same time, on the very top, you see a recording that shows the fast pyloric rhythm, LP, PYPD, up there. So you can see the difference in time between the pyloric rhythm and the gastric mill rhythm. Now, unlike the pyloric rhythm, the gastric mill rhythm is episodic. It's turned on when the animal eats, and it goes away for long periods of time. And so it's much more like your locomotory rhythms, and the pyloric rhythm, in a sense, is much more like your respiratory rhythms. Okay, now, a number of years ago, it was probably, 20-something years ago, Astrid Prince, who was then a postdoc in the lab, wanted to build a good model of the pyloric rhythm. And it's this study which really engendered all of the work that became our interest in climate change. So Astrid first built 1 million or so model neurons using sort of canonical voltage-dependent currents, including a sodium current, 2 calcium currents, 3 potassium currents, an H current. And then she took that connectivity diagram, and she took good candidate neurons for each cell type and hooked it together into the canonical architecture of the pyloric rhythm. And she then simulated all against all. She ran about 1 million simulations, and she chose the networks which gave what she called a good pyloric rhythm. And she found a number of very successful rhythms, and among them, I'm showing you here too, she found a number of good model pyloric rhythms, and then she was able to display, for these two in particular, you can see the underlying parameters that give rise to this rhythm. So on the left, you can see the AB on the bottom, the ABLP synapse was big. On the right, the ABLP synapse was small. In the left, the H current, that's the hyperpolarization activated inward current, HCN, was big, and on the right, it's small. And you can see those two sets of conductances are actually quite different, but yet the motor patterns that you see are very similar. So this was the first clear, at the network level, indication that we had highly degenerate structures that could do basically the same thing. Now, what this said to me, that any two crabs out there in the ocean, or any two people out here in the audience, didn't need to have identical sets of parameters, as long as they had a sets of interacting parameters that were good enough to produce the required behavior. So when I started talking about this, I talked. I remember one very interesting meeting where it was dominated by a whole bunch of very self-important physicists, and they said, well, this can't be true. Biology can't work this way. And I said, why not? And he said, well, we all know that biology is optimal and finds optimal solutions. And I said, since when are you optimal? And that shut him up. I was really pissed. You're not supposed to say things like that. So, but what those physicists said, and it was really important, and this is the comment that set off the next 20 years of my lab's career. And they said, it's easy to say that one set of parameters and another set of parameters can produce very similar behaviors, but if this is really true, then two animals with different sets of parameters out there in the ocean seeing different kinds of perturbations, you would predict them to respond differently. And that's absolutely true. There's no way the two sets of underlying parameters could give identical responses to all perturbations. And so they said, so it must not be true. And I said, no, what matters is we have to ask ourselves whether the perturbations that the lobsters and crabs in the ocean see, whether if they have different sets of underlying properties, whether they can respond in a stereotyped, proper way to the perturbations or the experiences that they're going to see in the natural world. So the question is, if these were really realistic recordings from two crabs, could those two crabs nonetheless respond correctly? So the first thing we did, and Dave Schultz and Jean-Marc Gaillard were in the lab at the time, was to ask ourselves, well, what were the underlying parameters in the animals? And Dave did the earliest molecular biology in my lab, and I don't know if I have that. Well, I have a later study in which, and here you can see recordings from 4 different PD neurons from 4 different animals, and these now are data from Dave Schultz's lab, mostly, from single neurons that we have hand dissected, put in tubes, and then have undergone either single-cell PCR for ion channel genes or transcriptomic measurements. And you can see, I think there are like 15 or 18 ion channel genes here. And if you're looking at an LP neuron, which you're not here, there's 1 LP neuron in each animal. There are 2 PD neurons in each animal. And you can see there's about a 2 to 6, 2 to 8-fold range of channel mRNAs seen in those unambiguously identified cells, all of which came from animals producing a perfectly good pyloric rhythm. So that was setting the stage. And then Dave and Jean-Marc did a bunch of voltage clamp measurements. And again, you see in the synaptic potentials and voltage clamp measurements a range of that 2 to 6-fold. And so if I walk around the world and I ask people about their data, this number, the 2 to 6-fold range, pops up in all kinds of preparations, all kinds of animals. I just was at a meeting in Europe where I heard a talk from someone recording in human cells, in slices from human patients. And again, the range of parameters that pops from that is, again, that 2 to 6-fold, 2 to 8-fold. And so I think that this is a really magic number. I don't really believe in magic, but this I do believe in, which is if cells or networks had to tune every parameter to 0.1% to get it right, that's obviously too hard for normal biological mechanisms. If things could vary 1,000-fold, you'd be hard-pressed to actually get a network that would work. But this 2 to 6-fold defines a manifold, a multidimensional manifold, which can define good enough behavior. And so these animals have to find solutions that sit on that manifold. They have to stay on that manifold if they're going to be good enough throughout their life. But they can move around on that manifold so that a spinal motor neuron in you doesn't have to have the identical number of sodium channels when you're 20 or when you're 50. You just have to be good enough. So that is the underlying premise. Now, the perturbations. This is where climate change came in. I'm thinking about, these data came from these crabs. These are all wild-caught animals, and they're old. They're 5 to 7 years old when we study them. So that means they've lived in the ocean. The ocean in New England varies from about, call it 2 degrees centigrade in a really cold winter to about 25 degrees centigrade in the summer. They can see 10-degree fluctuations in a day, and they just deal with all kinds of changing environments as the tides come, as storms come, as boats come, as everything that they have to deal with. So we were looking to test the notion. We were looking for global perturbations, and we wanted perturbations that in principle would influence every cell in the network. We did not want to do a targeted perturbation of a single synapse. That's a different experiment. We wanted global perturbations that would resemble the kind of global perturbations you'd see in the ocean. So we started studying temperature, the obvious one, high potassium, pH, and salinity. And we've worked at all of these. I'm only going to show you data from temperature, but Maru and Margaret Lee did some very beautiful work on high potassium. We have work with Dan Powell's lab on pH and salinity. So all of these things we were interested to see not only what each one of these perturbations would do singly, but how they would interact with each other. And you can ask Dan all of those questions. Okay, so we're going to start with temperature. And before doing that, I'd like to put this into your mind, because this is something I learned after I started getting invitations to meetings about epilepsy from the human neurological community. They saw the work we were doing on temperature, and temperature is something that the neurologists and many neurosurgeons are very, very concerned about as an environmental problem, because many seizure disorders, particularly Dravet syndrome, multiple sclerosis, the likelihood of severe stroke, human hyperkalemic periodic paralyses, all of these things, people with these disorders are very temperature sensitive. And basically the way you can think about it is if you have any impaired or partially impaired human nervous system, it will likely to be more temperature sensitive than a normal healthy control individual. So the kinds of events that are happening in the world and severe weather events have become very preoccupying to many neurologists, and you should understand that people with any neurological problem are at risk in the environmental perturbations. And then the last point just made me laugh. So I'll tell you, it turns out that rodent pups, when born, are about 25 to 26 degrees centigrade, and they're not 37 degrees, and they have to develop the higher temperatures as they mature over those first couple of weeks. And that's why people who work with mice use heat lamps and things like that to keep their little animals warm. And I found that really funny, because I got so tired of telling people that, vertebrates are so much better because they're temperature controlled. Not true. Not always true. Okay, now I just want to motivate in 1 second why temperature can be so perturbing to neural circuits. And this is just a slide that Max Kengot, who's here, made for me when I asked him to make me a slide like this. And what he's doing, this is a model of a Morris-Lacar model. It has a calcium current and a potassium current. And what he's done is he's got the same maximal conductances in the model, but he's applied a Q10 term to either the calcium current or the potassium current, where the Q10 is dealing with the temperature dependence of one or another of the biological processes. On the left hand, he's got a Q10 of 2 on the K current and a Q10 of 3 on the calcium current, and the reverse on the right. So you can see at 22 degrees, the two models are basically producing the same thing. In a sense, those are degenerate solutions. But as you go up in temperature, and on the left, you can see at 27 degrees, you have a depolarization block. You get a crash. As you go down in temperature, you have a more hyperpolarized crash, and that the effects of temperature on the frequency of oscillation are different than what you see on the right side, in which case the model just increases in frequency as the temperature increases and shows crashes in neither the 7 degree or the 27 degrees. So at some level, this is about as simple model as you can get and shows the diverse effects of temperature on behavior. Now, this was the first real set of data that we produced. These were data that Lamontagne collected in 2010, or he collected in 2006, and the paper was published in 2010. And you can see extracellular recordings from the pyloric rhythm at 7 degrees on top, and then at 19 degrees in the bottom. The pyloric rhythm is faster, not too surprising. What was very surprising to us, which is that the phase relationships, the relative timing of those elements, was maintained over that entire range of temperatures. And so that says maintaining constant phase is not trivial. And so it said these animals had found a set of solutions that allowed them to maintain phase, because in a motor system, the relative timing is much more important than is the frequency. You know, I can walk in a number of frequencies, but to walk, I have to have extensors and flexors go in the right order. I'll skip this. This just shows that intracellular recording is the same thing. But what Lamont noticed early on, and this is going to be very important, is that if he increased the temperature beyond a certain point, he started seeing crashes. And here are 4 sets of crashes from 4 different animals. Notice the dynamics are entirely different. And that's exactly what you'd predict. If you're starting out with 2 or 4 animals with different underlying sets of parameters, you'd expect them to crash with different dynamics, and that's exactly what happens. So this, again, verified the idea that all these animals could behave fine at the temperatures they would normally see, and then crash and reveal their underlying differences. So now I'm just going to show you one slide from work that Dan Powell did. This slide is much easier than it need to be, but I need this slide for later. And these are now recordings from what we call a particular form of the gastric mill rhythm. And up there on top, you can see the slow gastric rhythm and the fast pyloric rhythm. The pyloric rhythm is on the PDN. The LG and DG show you the gastric mill rhythm. First at, I don't know what the temperatures are. I think it's like, does it say on the slide? It must say somewhere. It does. It's like 7 and 20 something. And notice that both rhythms increase in frequency and both are maintained, and they maintain. The reason why we really like this work is because this rhythm maintained a very peculiar property it has, which is a particular coordination between the pyloric rhythm and the gastric rhythm. Okay, now the segue to climate change. 2012. I'm sitting in my office, and I asked Sarah Haddad, who was then a beginning graduate student, to repeat Lamont's data, and she came back and she said, "They're not crashing." That's her exact words. And I said, "What do you mean they're not crashing?" She said, "I've got the dish up to 35 degrees, and they're not crashing." And I said, "You're not measuring the temperature right. You're doing something wrong." She came back. She got really angry at me, and she said, "They're not crashing. Look, it's going 7 hertz." And I said, "Oh my God." I'd never seen a pyloric rhythm at 7 hertz. These are her data, 11, 20, 25, 30, 36 degrees. And then later, this was, I think in March of 2012, and then we later learned, talking to the people down at Woods Hole, that that was the year the winter never was. The crab habitat was 6 degrees warmer than normal. The guys at Woods Hole said the squid all went to Maine. The dogfish all went to Maine. All the animals were in the wrong place. And when they wanted to collect things, they had to go to places they'd never been to get animals. So when I presented this at a FENS meeting, I had a graduate student come up to me with tears rolling down her face saying, "Oh, climate change is real, climate change is real." And I said, "Yeah, we'll wait till next year." So anyway, following years, the temperature went back to normal. And things went more or less back to normal. Or more normal. And then Marder was doing some experiments in 2021. And here we have some of Mara's data from 11, 15, 19, 23, 27, 31, 35. Again, you can see the strong temperature dependence and the lack of crash at 35 degrees. And then Mara, that's just plots of those data. Mara, being very sensible, went back to our lab notebooks and data, and she mined data going back from Lamont through Sarah and then Jacob Ratliff and her data. So up there in green, you see Lamont's original data from 2006, where at 11 degrees you have a nice pyloric rhythm, at 21 you have a nice pyloric rhythm, at 31 it's completely crashed. 2012 was Sarah's data. Again, that's the no crash at 31 to 35. 2016 was Jacob Ratliff, another student who was in the lab at the time. At 31, his networks were entirely crashed. And then Mara's data, 2021, you can see 31, everything's fine. 39 or 35, whatever it is, it's not. But then Mara went to the next step, and she went and did something which may not be possible for somebody in my lab to do again, which is she went to the NOAA website, and she got the data from NOAA about the temperature on a buoy 16 miles off Boston Harbor, and she plotted the average water temperature in all those years, and the colored dots match the data. So first of all, you could see both the average summer and the average winter water temperatures are going up, and also there's a correlation between the physiological data in the lab and the water temperature that these animals had been seeing. So this really sort of frightened us because it said we're actually seeing the direct consequences of those data. And the thing that really disturbed me the most, and which is a separate issue, I've highlighted. Now I highlighted, you can see I don't do very well, you know. If Mara had done this, it would have been neat, but I did it. I highlighted the 11 degree data because 11 degree has been our classic control temperature. And if we had recorded only at 11 degrees, we never would have known the fact that these altered temperatures would have changed the way the preparation would respond to other perturbations. So these are what I call cryptic changes. We see them in response to many perturbations, and these are the changes that I think we should be most concerned about, the changes that are not revealed, that store the history of what the animal has seen, and you only see the responses altered by those changes in response to a later perturbation. Okay, now, so degenerate circuits can hide cryptic differences due to differential history that can alter and shape future responses to additional challenges. So, neuromodulators can extend the range over which circuits can function at extreme temperatures. We've done some work about this, as has Wolfe, and as did Scott Hooper many years ago, and it's very clear that some modulators can actually extend the temperature range at which networks can function. I'm not going to show you that, but to go back to what the role of this meeting is really about, is for the future, getting to the biological mechanisms underlying short and long-term adaptations and acclimation. Now, long-term animals, such as ours, see seasonally and yearly changes in their environment. We know that changes occur in all tissues because the acclimation people have been studying everything you could imagine has been studied in various animals, and nothing has been studied in a very organized way. But there are changes all over. And anything that anybody ever studied has changed during acclimation. But what intrigues me as a problem going forward are some environmentally induced changes rate limiting. Do some tissues respond earlier than others? How are multiple changes at different parts of the animal correlated and coordinated? And are there global sensors of function, or does each tissue take care of itself? Where are the control mechanisms? And then what molecular biochemical mechanisms keep all body parts functioning together? because that's what matters. And if I have a couple more minutes, I'm just going to show you some very recent data, which is still preliminary, but that Kathleen Jackery has been working on. These are some of the stomach muscles, and Kathleen's just colored them in different colors. The muscles innervated by the LG neuron, that's part of the gastric mill, are GM5, GM6. And down here, P1 is innervated by the LP, CPV1 by the PD. And what Kathleen's been doing is she's been stimulating the nerve and recording excitatory junctional potentials, the synaptic potentials in the muscle, that's the functional output, in response to various stimuli. So here are data from GM5, GM6, GM8, CPV1, CPV4. There are 40 sets of striated muscles that move the stomach. It's a very complicated device. In all cases thus far, if you recorded the neuromuscular junction, and people had done this 40 years ago and seen this, there's a hyperpolarization of the resting potential. The EJPs get smaller, the conductance increases, and this is very canonical. Now, this, as I said, was noted a very long time ago, but Kathleen put her head to, and that just summarizes the data in these different muscle types. But what Kathleen has done is take it one step further and to try and figure out what the biophysical mechanism of this is. So what Kathleen did is she's done IV curves at 11 degrees and IV curves at 21 degrees. And where those two IV curves intersect is the reversal potential. That reversal potential is basically the equilibrium potential of the ion that's carrying that difference current or that. So under control conditions, you can see there's an intersection at, I don't know what it is, wherever it is. And then if she shifts into low potassium, thus shifting the potassium equilibrium potential in the hyperpolarized regime, that intersection point shifts down. In high potassium, it shifts up, and those are the data. So the response of these 11 to 21 degree shifts tells you that the current that's responsible for this hyperpolarization and increase in conductance is a potassium current. And our current hypothesis is these are two pore potassium currents. And these are data that come from large databases from Dave Schultz's lab, where he's pulled out in the LG, in some of the neurons, you see a channel he calls KCNK1, which is found in high concentrations, and then there's something he calls KCN2, and these are homologous to 2-pore K channels in humans. And what we're currently trying to figure out is how to actually figure out exactly what those genes and those channels are. Now I'm just going to end with a very pretty set of recordings that an undergraduate, James DiMartino and Kathleen, have done measuring muscle movement. So on the top, you have the impulses that James has applied to the nerve innervating the muscle, and he's put the muscle on a movement transducer so you can see the movement. And at 11 degrees, in this given recordings that Kathleen pulled out of Dan Powell's recordings from the LG neuron in his paper on the effects of temperature on the gastric mill rhythm, given those patterns of activity and now measuring at 11, 13, 17, and 21, you can see the muscle follows at 17. It's no longer really able to follow very well. It's completely collapsed to 21. However, when James uses the neural discharge that Dan measured at 21 degrees, he sees, obviously, a much faster rhythm. The muscle follows, and it doesn't completely crash at 21, and it's still working pretty well at 17. So this is telling you that the effect of high temperature on the nervous discharge is actually maintaining the muscle movement to a greater degree than it would be at a lesser muscle discharge. So this is all still beginning days, but this goes to the issue of whether or not you can start taking apart the different components of, in this case, our neuromuscular system, and try and ask which components are most temperature sensitive or most pH sensitive or whatever, and are there mechanisms by which animals can protect themselves against the deleterious effects of temperature by employing a lot of other mechanisms. And I'll just stop there. I'm not too much over. Thank you.
We have some time for just one or two questions. Carlos?
Wondering if it might be at the order, but I was wondering if you could give us advice of like how to apply all this, the degeneracy principles that you've been talking about. Networks with very different architectures make outcomes, and I was wondering if you have any advice for people that are interested in comparative work as we accumulate more knowledge about more species, get more informed and more.
I think you have to be really careful about deciding what features you care about. And that's where your biological intuition comes into play. A random computer scientist might say, I care about feature X, and that might not be a feature relevant to the animals in the world. So I think where that interdisciplinary collaboration is most important is to make sure that all those analytical and statistical tools are coupled to really good biological intuition. And I don't think you get that from a random mathematician, physicist, or computer scientist unless they've been hobnobbing with biologists. So we've done all sorts of things and gotten lots of non-intuitive, you can get a lot of non-intuitive data, but I think it's a real problem. I think that people force data into all sorts of statistical models that may not be relevant to my crabs in the ocean. My crabs in the ocean do care about whether they're eating or not. They care in my tanks too. They change in many, many ways. I don't know if that helps. We can talk later. Yeah.
Maybe, and this is smart, but do we know if the parameter space that you're talking about either expands or contracts under elevated temperature?
So you don't want to talk about the amount of pieces of tissue that we have in our freezers, because obviously what we've been doing in the last year is we've actually started both keeping tissues, lots of different tissues from animals collected at different times of the year. We now keep really, really good records for every animal, when it was collected, how long it's been in the tank, what temperature it's been at. And then we started to do some real acclimation experiments. And again, when we do that, we keep a bunch of tissues. And then we're in the process of sending them to my dearest friend, Dave Schultz. And we want to see whether or not our time of year and our laboratory acclimation produces serious changes in gene expression as well as the behavior. It's a non-trivial problem, because what we've been trying to say, okay, if we keep tissues from 5 or 6 different places, muscle and nerve and heart, maybe we could ask this, do those things all change in a correlated, concerted fashion? Again, that's brute force. You have to have a friend like Dave.